Hello students, welcome to Legacy IAS Academy. Today we are going to discuss about the recent topic in news, that is the genome editing technology. We are going to discuss what does this term mean, and also we are going to discuss that how genome editing technology is different from genetic modification or genetically modifying technology. So first of all, the context in which we are discussing today's issue is because on the Wednesday, that is 2 days ago, the central government paved the way for a much easier introduction of genome edited crops. And this has come when there has been a clear distinction that has been made by the government and the expert committee between genetically modified crops and genetically edited crops. And it is since the government has clarified that genetically edited crop is different from genetically modified crop, it have put a very easier norms for their introduction into the market and introduction into the food chain of food chain. So let us try to understand what is genome editing. So if we try to look at the background, somewhere earlier in the beginning of 19th century, the German and American scientists have discovered a way by which they can actually cut the DNA strands and edit the genes. To give you a brief background, DNA strands are made up of two strands which are arranged in a helical fashion. These are two different nucleotide chains which are bound by two different nucleotide bases. Basically, we have arrangement of four nucleotides, that is adenine, then we have guanine, then we have cytosine, and then we have thymine. So these four bases are the base pairs which are arranged between these two nucleotide chains. That is how the general structure of the DNA looks like. So what these scientists discovered, that actually this DNA can be cut. For example, if we have this is one nucleotide and this is the other nucleotide basis, we can make a cut at somewhere in the central part of this, and then we'll have, instead of a single helical structure, we'll have two different semi-helical structure. This achievement or this cutting of the DNA can be done by two different enzymes, that is the site-directed nucleases, also called as SDN in short, and we have sequence-specific nucleases, that is called as SSN. So nuclease is nothing but basically it is a kind of enzyme which produces a cut in the structure of DNA. SDN is directed for a particular site. For example, by employing SDN technique to edit the genomes, scientists can target any particular region of the DNA and that nuclease will make a cut here. While you talk about the sequence of specific nuclease, they make a cut depending on how the sequence of these base pairs are repeating in the DNA. So we can target the specific base pair and the nuclease or enzyme can make cut as per that particular base pair, creating a, we can say, repeated pair of AT bases or repeated pair of GC bases and similar like that. So basically, if we try to understand it from the diagram, what we can see from here, that this is the fully developed, we can say this is the full structure of DNA, semi-helical structure of DNA, as discussed with two bases and two this chain. Now after making cut, this nucleobases has been cut from the central part, and now we have a single structure of nucleic acid. Apart from that, the more recent development that has happened in this particular sector is that we have development of a CRISPR associated protein based system. CRISPR stands for clustered regularly interspaced palindromic repeat, and this is the most advanced system which can target a specific part of DNA and make a cut according to the demands, according to the desire. And that is why it is used for a targeted intervention of any particular genome sequence. The main advantage of this particular genome editing technique is that it can be used to edit genome so that specific traits can be entered into the gene sequence. For example, especially if it is done for the plant varieties, what scientists focus on, that they want to provide the plant with, or the plant DNA with certain kind of qualifications, to certain kind of traits that are not naturally present in the plant. For example, you talk about BT cotton. Now BT cotton is a kind of genetically modified crops, and it has been inserted into certain traits that the cotton plant itself fight against any kind of roundworm or any kind of other kind of infestation like pink bollworm infestation and something like that. So that is the most important significance of genome editing. Now the process of genome editing can be carried by three major methods. It is called as SDN1, SDN2 and SDN3. So let's try to understand about these in brief detail, that what does SDN1 refers to. So SDN refers to that method, SDN1 refers to that method where the scientists try to change the host's genome's DNA by a small insertions or deletion. So what is insertion and deletion? Now, insertion refers to a method of inserting certain nucleotide bases that were earlier not present at that particular site of DNA. While deletion refers to actually erasing the nucleotide bases that were earlier present. So insertions and deletions can be used to modify or to change the overall structure and the overall trait and specificities of the DNA. However, one thing that is to be ensured in SDN1 process, that no foreign genetic material is introduced in this particular process. That means if scientists are able to cut the DNA and then insert or delete certain base pairs, then the body's mechanism or DNA repair mechanism will work in a similar way how it would have worked by some natural damage caused due to the DNA. Now coming to SDN2, SDN2 is using of a very small DNA template to generate specific changes. However, here also it not have any kind of involvement of alien or foreign genetic material. That means both SDN1 process and SDN2 process can be look very similar to the earlier process of hybridization that was done before the genetic editing or genetic we can say modification was actually understood properly. Now the third process is something that is called as SDN3, and here we have involvement of a much larger DNA elements as compared to SDN1 and 2, and or many times even full length genes of foreign origins are introduced into the DNA structure, and it is very much similar to genetically modified organisms development. So SDN1 and SDN2 are relatively much liberally, or we can say much less regulated, because their threat to the environment is relatively lesser. However, SDN3 is regulated in the similar way how we have a regulation of genetic modification in different countries of the world, because the risk it poses to the environment as well as the other organisms are relatively higher. Now, we briefly try to look into the difference or comparison between gene editing and genetically modification. Two major things are there. First, what we can say roughly, that in gene editing, the foreign genetic materials are not introduced, while in genetic modification, the introduction of foreign genetic material is the most important things. Second, gene editing can be understood to be an alternate to the selective breeding that was done earlier time, and when the gene editing, gene modification was not understood. While in genetic modification, we scientists basically try to understand that what kind of traits they want to impart to that particular organisms, whether it is plant organisms or animal organisms. And for example, we can understand this from the genetically modified cotton plant, which is grown at a very large extent in India. So what scientists have done, they have found a bacteria that is commonly found in the soils of India. So this bacteria is called as Bacillus thuringiensis, and this bacteria has yielded two separate genes, that is called as Cry1AC and Cry2AB1, and these genes has been introduced into the genetically modified into the cotton crops that has made it genetically modified. And the introduction of these two genes from the Bacillus thuringiensis has provided the cotton the ability to fight against the pink bollworm infestation. So that makes the cotton crop much more resilient against pink bollworm infestation, and that increases the crop yield and increases the overall value of the crop produced in any particular part of India. So that is a major difference between gene editing and gene editing. Now, the regulatory issues obviously are the genetically modified organisms and genetic modification process as a whole, and that we can understand because in India till now only BT cotton is the one genetically modified crop which is grown in a large scale area. Other example of crop, especially food crops, for example BT brinjal was there or GM mustard was there, they have not been able to, the government has not been able to actually introduce them into the larger market structure because of severe protest that has been done by both scientific community as well as the farmers organization. So the three most important issues or concerns regarding genetic modification is that there is a biosafety issue and we have incomplete data to understand the full scale implication of introduction of GM crops into the environment. Second, genetic engineering appraisal committee, that is the we can say the main body that decides about the introduction or about giving a regulatory approval for the introduction of GM crops, they have also said that these crops are susceptible, or the we can say the genes that is present in this crop or the modification that is present in the particular crop are much more susceptible, that they can even target the non-target organisms, and once they start spreading in the wild environment, the overall effect will can be much more disastrous than understood properly. So it is due to these reasons, due to lack of information, lack of knowledge, and lack of we can say practical evidences, we are not able to, or the government is not able to introduce these crops at a large scale basis, especially the food crops. So what we can say in conclusion, that as far as the genetically edited crops are concerned, or genetically edited organisms are concerned, the signatories which were called by the government of India, who are mostly scientists having Padma awardees, they have categorically said that the variant developed through SDN1 and SDN2 technique do not have any alien DNA, as discussed before, and since as they do not have any foreign DNA, alien DNA, they can be treated as simple hybrid crops and can easily begin approval to be introduced into the market. And also the government has agreed to their proposals and has said that it would rely on the reports of a special committee, that the government is going to institute a institutional biosafety committee to exclude exogenic genetic materials that might be introduced in the genetically edited crops, and thus it is the whole process of giving regulatory approval and giving the market access will become much less cumbersome and much less time consuming, and thus genome edited crops can be easily introduced into the market, and thus will help both the farmers, consumers, as well as the government at large. So that is all about this particular issue. I will come soon with another interesting topic. Till then, take care. Thank you very much.